Hey y'all. So we're back with another chunk of chemistry. I'm done coughing. If you were watching the previous video, I had a tickle in my throat. So let's talk functional groups and those biological macromolecules. So these are the ones you need to know. Hydroxyl, remember, was, do I have like pen capabilities? I don't even know. But it was OH minus. We can probably skip carbonyl. Carboxyl is COOH. Carbonyl is CO, if you're wondering. Amino is NH3. And then the phosphate group, we kind of always just drew it as like a P with a circle. But they're going to come into play as we talk about like different types of molecules. So that you don't just have to memorize them, but you should be able to recognize like within something what's going on. So let's kind of talk about macromolecules. So these are all carbon based. Carbon has 4 spots in its valence shell that are open. So it kind of equally gives, takes, and shares. It bonds really well with everything. Remember that a monomer is kind of a singular thing when you're talking about a molecule, and then a polymer is a chain of monomers. So a polymer is like a pearl necklace. I'm trying to think if I have any examples that are here. Not really. And a monomer is kind of like that singular pearl or that singular bead. So keep that in mind. So let's talk about these 4. These carbohydrates, proteins, nucleic acids, and lipids. So I did mention that monomers are kind of that singular entity. Remember that monomers are joined together by a reaction called dehydration synthesis. So dehydration refers to the removal of water. Synthesis refers to like putting molecules together, okay? So we remove this water here and we're going to link these monomers together. The opposite of that then is adding water in to split. So the water part, the adding of the water part is hydro. Splitting is lysis or lyse. And so hydrolysis then, oh, my daughter has a band lesson. If you can hear her clarinet, excuse that. But then adding that water in to split is going to break apart those polymers, okay, by adding water. So carbohydrates, I'm going to put everything up on the slide. We kind of have some categories. So the monomer for each of these, like I believe this is a chart that we had made way back when. The monomer is a monosaccharide. Saccharide means sugar, so we're talking simple sugars here. The one you should be familiar with is glucose. Obviously, that comes into play later when we're talking about cellular respiration and photosynthesis and stuff like that. The polymer then is a polysaccharide, so that's kind of nice. There are disaccharides. I don't think there's any memorization that needs to happen with that. The bond is a glycosidic linkage, and the job of carbohydrates is either energy or structure. Okay, there's 2 jobs, energy and structure. And so kind of our 2 energy ones that you should know, the 2 polysaccharides are starch and glycogen. So you've got glucose in your blood. You want to store it somewhere for later, and animals are going to store it as glycogen in our liver, glycogen. Plants are going to store it as starch, oftentimes in their roots. That's why like root vegetables, like carrots and stuff, are sweet when you cook them. In terms of structure, the ones you should know are cellulose, which make up the cell walls of plants, and then chitin, which make up those exoskeletons of insects. They're both really hard. Oh my God. Lipids, which a lot of us just use interchangeably as fats. They are all monomers. There's no like polymer form of lipids. They're also hydrophobic, so they do not like water. And we should look at ester linkages as the bond there for fats. So the main function is energy storage. So if you look at like per gram, a 1 gram sample of carbohydrates has 4 kilocalories. I'm like accessing files from September in my head. And a 1 gram sample of a lipid has 9 kilocalories. So there's more CH bonds there, which means there's more energy when you break those bonds. So you had a question on a quiz like a million years ago, seems like so long ago, that talked about like why is it more efficient to store our energy as fats? And it's because it takes up less room. If we stored our energy is carbohydrates, we'd be twice as big. Okay? So fats in general, you have a glycerol and then 3 fatty acids. You should be familiar with saturated and unsaturated fats. Saturated are all the S's. They're solid at room temperature. They have single bonds. We're talking things like butter. And they're straight. That's another S. Unsaturated have at least one double bond in that hydrocarbon chain. They're bent or they're kinked at some point. They're liquid at room temperature like oils. Another example of lipids that you should be familiar with are phospholipids. That's going to be coming up when we talk about cells as well. That's going to make up our cell membrane. I have that lovely model at school that has the heads and the tails, the hydrophobic tails that face inward, and then the hydrophilic heads that face outward. Okay, so that makes up cell membranes. And then steroids, I think you should just be familiar with like that they exist because we didn't really ever get to hormones. They took that out of the curriculum. Good. Proteins, what do? They do everything, which is what I think you would tell me. So you should be familiar with the structure of an amino acid, which has one end has this carboxyl group, this COOH. The other end has this amino group, this NH3. And through dehydration synthesis during protein synthesis in a ribosome, when the tRNA brings the amino acid, that's how they're going to join. Okay, so you should be able to recognize that in an FRQ. That bond that's created is called a peptide bond. It's strong, it's covalent. Okay, so enzymes are coming. We'll talk about enzymes. But when you talk about protein structure, you should be familiar with the primary, secondary, tertiary, quaternary. The actual order of the amino acids is your primary. The alpha helix and the beta pleated sheets, that's your secondary. Tertiary is 3D. So the third level is your 3D folding, and then the quaternary, there you go. It won't move. The quaternary then is when you're looking at multiple polypeptides interacting with each other. And now when we change the shape of that protein and it can't do its thing anymore, that's what we call denaturing. And that was one of our go-to answers if you're not sure what to write, which I'm sure you remember is denaturing. Good. And then finally, nucleic acids. Nope. Nucleic acids have that nucleotide as its base unit, as its monomer that consists of a sugar, a phosphate group, and a base. Those bases are interchangeable, not interchangeable. They can change. Adenine, thymine, cytosine, guanine for DNA, and then replacing that thymine with uracil for RNA. Then when they get strung together, that's when we call it a nucleic acid. The only two nucleic acids are RNA and DNA. Mr. Morelli will be doing a sequence of videos for like all that genetics type stuff. Their job is to store and pass on hereditary information. Okay. So that's kind of it. We're going to try to keep these videos short and like topic based. The next one's going to talk about the cell. Peace.